Hi everyone, this is Miss Moore and today we are going to learn about genes and chromosomes. This lesson builds extensively on the lesson we had previously on DNA structure and function. So we're going to carry a lot of those concepts into today's lesson, starting with a couple warm-up questions to refresh us on what was really important from last time. What is DNA? What is it made out of? And what is the central dogma of molecular biology? Please pause the lesson video here while you do your warm-up questions. DNA is like a molecular blueprint for a living organism. It contains all the genetic information required for that organism to survive and reproduce. It's a long chain-like molecule shaped in the form of a double helix and composed of nucleotides. Tiny molecular structures composed of a sugar, a phosphate, and a nitrogen base. The central dogma of molecular biology is that DNA makes RNA makes protein in a process called protein synthesis, which we'll be looking at in detail in today's lesson. So far, we've been casually throwing around the word gene. Genes, genetic information, genetic code, genetic instructions, and we've been kind of intermingling the word gene with the word DNA. So before we continue, we need to clarify the difference between DNA and genes. Human DNA contains over 3 billion nucleotides and measures about 1.8 meters in length. This is pretty long, and all of that is stored inside every single one of your cells. Each long strand of DNA contains the instructions for making over 100,000 different proteins to support all your body functions. These instructions are neatly organized into groups along the strand called genes. Therefore, a gene is a small section of a DNA strand that codes for a specific protein. Human genes are usually around 27,000 base pairs in length. By base pairs, I'm referring to the nitrogen bases, adenine, thymine, cytosine, and guanine. Genes that code for more complicated proteins can be up to 2 million base pairs long. Here's an interesting fact though. Among multicellular species, like you and I, not bacteria, the complexity of the organism, like if we compared ourselves to a newt, I'm pretty sure you would say that humans are a more complicated multicellular organism than newts. The complexity doesn't correlate with the number of base pairs. A human has 3 billion base pairs of DNA, while a newt has 15 billion. This diagram shows the difference clearly between a DNA strand, which is the whole long genetic sequence, and a section of it, which codes for a specific protein, like in this example, the section for making hemoglobin, or this section for making hair. Now, last year in Science 9, we spent a lot of time studying cell division, like mitosis and meiosis, and so two words that were being used frequently were chromatin and chromosomes, and we were also using these words interchangeably with the word DNA, but they're actually not the same. Most of the time, DNA can be found loose in the nucleus. By loose, I mean not coiled up, basically like a pile of spaghetti. And each strand of DNA, like a spaghetti noodle, is curled around a special protein called a histone, which helps to organize the DNA. Think spaghetti and meatballs. The spaghetti part is the DNA, and the meatball part is like the histone protein. That's kind of a crude analogy. It's a lot more specific. It kind of looks like this, with the DNA strand coiled in a very specific way around the histone protein. This part of the diagram shows the chromatin. So if you picture a big pile of that, you can kind of see where I get the spaghetti and meatballs analogy from. When it's in this form, we call the DNA chromatin. It's this big pile of genetic material composed of DNA and histone proteins. DNA spends most of its time in this form. The only time when DNA is not in the form of chromatin is when it's in the form of a chromosome. Cells frequently divide to form new tissue. During cell division, like we learned about last year, the DNA curls up into a more compact form. This makes it more efficient for the cell to segregate the DNA during cell division, which means give one set to one new cell and another set to the other new cell. In cell division from last year, you might recall a single cell splits into two new daughter cells, and they each have a copy of the DNA. So, in order to prepare for cell division, the cell coils all of its DNA into tightly coiled bunches called chromosomes, which is pictured here in the diagram. Below, I've given you guys a diagram of mitosis, which you'll probably remember from last year. You don't need to learn all the steps of mitosis over again. All you need to know is the cell spends most of its time in interphase, which isn't really part of cell division. It's the part of the cell's life where it's performing all of its regular cellular functions, growth, body functions, protein manufacturing, and all that sort of thing. Then from prophase to telophase and cytokinesis, the cell divides, producing two daughter cells. During prophase, the DNA is coiled from chromatin into chromosomes, and that's how it exists for the remainder of cell division. Until cell division is over, the DNA uncoils itself back into the form of chromatin. Human cells normally have two sets of chromosomes, one inherited from each biological parent. There are 23 chromosomes in each set, so in total you have 46 chromosomes in each of your cells. Each set consists of a pair of chromosomes of the same size and shape that contain the same genes. The chromosomes in a pair are called homologous chromosomes. Those are pictured right here and also here. I really like this diagram because it clearly shows the difference between a strand of DNA and a chromosome. It also shows that a section of the strand is a gene. Here's two examples for you to try on your own. What are genes and how are chromosomes different from chromatin? If you have some time, I really recommend this video on YouTube titled, "What is a Gene?" It explains it in great depth and provides some really interesting examples of what your genes can do. Please pause the video here while you try these examples. Okay, let's go over our answers together. Genes are small sections of the DNA molecule, usually around 27,000 nitrogen base pairs in length, that code for a specific protein. Chromatin is a mass of genetic material consisting of DNA strands wrapped around histone proteins. Kind of looks like a pile of spaghetti and meatballs, like this. Whereas chromosomes are tightly coiled structures, tightly coiled collections of chromatin that are much more efficient in this form when it comes to separating the DNA during cell division. So now that we understand all the parts of DNA and all the forms we can find DNA in, let's get on to how our DNA provides the instructions for proteins. This is a process called protein synthesis, the production of proteins. Proteins are long molecules made up of chains of amino acids, as we saw in our last lesson. Amino acids are the building blocks of proteins. Depending on how you connect them, you get completely different proteins. And there are 20 different types of amino acids that can be joined together in so many different configurations. The genetic instructions in our DNA dictate the order in which these amino acids are assembled to make proteins. Discrete sections of the DNA molecule, which we now know are genes, code for specific proteins by telling the cell how to connect the amino acids. That's why living things on Earth display such a wide variety of traits. It's all the different ways you can build proteins. Changing the order of just one pair of nitrogen bases on a strand of DNA results in a new gene that determines a completely different protein. For example, if we took the sequence ATT-CGG and we just switched the first two nitrogen bases so that now it goes TAT-CGG, this differs in the order of only two bases, but it could lead to such a significant change, a different trait being expressed, such as your eyes being blue instead of green. In fact, most mutations are actually just a small change in the order of nitrogen bases in your gene that leads to a different protein being produced than what was perhaps intended. If you have time, I strongly recommend watching this video on YouTube called Protein Synthesis, which goes through the entire process in a very visually dynamic way. For now, on the next page, I'm just going to summarize with you the important stages of protein synthesis, and we're going to rely on this diagram to do that. So the diagram is labeled stage 1 through 6, and below the diagram, we're going to detail what's happening in each important stage. The first thing that has to happen up here in the nucleus of the cell is that you have to rip open your DNA, which is called unzipping. And you'll see in this image here, this is a fully zipped section of the DNA, and this here is our unzipped section. As soon as you unzip it, you can start copying it, which is what happens in step 1. A molecule of DNA unzips between paired nitrogen bases. In order to do this, you have to break the connection between the nitrogen bases, which your cell has the components to do. Then each strand of DNA directs the production of a complementary strand of RNA, which is a nice way of saying your DNA is copied into a different molecule called RNA. And this is actually a perfect copy of 1/2 of your DNA strand, which is all that's really needed in order to code for a protein. The only difference between RNA and DNA is that, well, RNA is single-stranded since it's only 1/2 of a DNA strand, but also RNA uses a different nitrogen base because it doesn't have thymine. It uses a base called uracil, which we symbolize as capital U. Once this copy of the genetic code is ready to leave the nucleus, we call it messenger RNA, or mRNA, because it runs the instructions from the nucleus out into the cytoplasm. In step 2, the mRNA carries the genetic information to the cytoplasm, which is the interior of the cell where protein synthesis takes place. You can see that in the diagram here. Transport to cytoplasm as the strand of mRNA exits the nucleus. In step 3, the mRNA arrives at the ribosome. This organelle is the site of protein synthesis. Here, the mRNA is fed into the ribosome and read by a helper strand of RNA called the transfer RNA, or tRNA. In the diagram, you can see the strand of mRNA being run through the ribosome in one direction. As this is happening, the ribosome is reading the bases on the strand and telling the tRNA which amino acids it needs. The tRNA then fetches the required amino acids. When it has the correct one, it docks on the ribosome and assembles the amino acids based on the gene sequence being read off the mRNA. This is step 4. Here you can see a molecule of tRNA arriving at the ribosome and then docking and transferring its amino acid to the ever-growing strand, which will form the protein. Finally, in step 5, the amino acids are assembled in a specific order to form a new protein. In our diagram, you can see the growing chain of amino acids here in step 5. Step 6 isn't anything fancy, so I left it off the description. Basically, the tRNA undocks and leaves the ribosome when its job is complete. Okay, everyone, that's it for today's lesson. It's time to work on your assignment. Please don't forget to check your solutions before turning in your work. Bye for now.